Thank you very much, Godse ma'am and Sejal ma'am, for brief introduction. Uh, it's an honor and a pleasure to deliver a talk today to our own institute, where I did my diploma degree and masters, and and it is also a great thing for me to deliver this in front of Asha ma'am and Maruti sir, who have taught me when I was a student there. And so I will just start with my topic now. Okay, can you see these slides?
Yes, sir.
Okay, so the today topic of my presentation is influence of natural excipient for solubility enhancement and supersaturation maintenance using hot melt extrusion and spray drying. I will do a case study of hot melt extrusion and spray drying. These are my contents. I will just shortly give you an overview of introduction of my topic and why we are doing this. Solubility enhancement, then the physiochemical properties of the polymer, then the ASD manufacturing, then the evaluation of the amorphous solid dispersions, and the conclusion. Introduction, what is the current challenge? The current challenge what pharmaceutical companies are facing is the new chemical entities which are evolving. They are having poor solubility and poor permeability. This belongs to the class 4, while the solubility, the poor solubility and high permeability drugs belong to the class 2 drugs. And the objective is to improve this solubility limited bioavailability. Then to do this, what can be done is use of a polymer which can help to improve its solubility and maintain supersaturation. For this purpose, in our working group, the idea came up where using HPC, SSL, and HPMCAS, which is a pH dependent polymer, and HPC is a pH independent polymer. To combine those and achieve the site specific delivery, then we came up with an idea, why can't we use a natural excipient then, which can solve this purpose? So my PhD topic was focused on this, where I try to use several natural excipients. And with this, why we want to do this is to extend the chemical set of the existing synthetic polymers. And the natural polymers are easily resourceable. They are available widely in nature. And they are only the remaining set to evaluate as a matrix in amorphous solid dispersion. Then there are different techniques to enhance the solubility of the poorly soluble drugs, which is the most challenging job at the moment. The first technique is by solid dispersion. The second is the silica technology. And the third is the API technology. The solid dispersion, this is the physical modification of an API substance. You can convert a crystalline substance to an amorphous form using hot melt extrusion, spray drying, freeze drying, or spray freeze drying. Hot melt extrusion and spray drying are the industrially applicable and widely accepted techniques. And that's why the focus of my topic was to evaluate those techniques and see their performance of the product on each other, the process performance. And the silica technology is also quite new at the moment, where mesoporous silica is widely used, Siloid 244, and then also Aeropol from Evonik. And the API technology is more of a chemical modification by using salt formation or co-crystal. Then the marketed ASDs currently, you can observe that hot melt extrusion and spray drying, they are widely used and widely accepted in pharmaceutical industry. Right from 2013, you can observe every year there are new approved new chemical entities. Noscofil, the product name was the brand name from MSD. They use HPMCAS for spray drying, and by method of preparation was spray drying. Well, the AbbVie make Ombitsaver and ritonavir combination using copovidone polymer and by hot melt extrusion. Then the Sobospovir and Veltavasir is also widely known for hepatitis B virus treatment, and it is prepared by the method spray drying. While you can see the recent was from 2018, where doravirine was prepared by spray drying technique, and this lamivudine, tenofovir, and were granulated separately, and then they mixed together. So doravirine was very poorly soluble. That's why MSD thought an idea to make a spray drying and then combine with the granulated form, so and make a tablet formulation out of it. The goal of my study was to investigate the effect of various ratios of shellac. So one of my natural excipient, which I will try to focus on today, which can be used as an additive in amorphous solid dispersion by spray drying and hot melt extrusion to form a single glassy solution. Then to investigate the impact of using the shellac on solubility profile, that is solubility enhancement or improvement, and supersaturation maintenance. Then I will introduce the API, what I am trying to, as my model drug. It's loratadine. It's a weak base having pH dependent solubility. The solubility you can see at pH 1.1 is 0.168. So which is, we can say, soluble at lower pH, but as the pH increases, the solubility drops. Then that's the problem, that when you take an API medicine, it gets dissolved in a stomach pH, but when it goes to the intestinal, it gets precipitated and it is no longer available for effective action. So we need to address this problem, and that's why we did some physiochemical characterization, where the melting point observed was 134 degrees Celsius and the glass transition temperature was 34.3 degrees Celsius, making it excellent candidate for hot melt extrusion and as well as spray drying. And shellac is a well-known excipient in India because it is mostly common in Asian countries and especially in India. So shellac is not an excipient, but it is a complex mixture of organic acids containing aluritic acid, shellac acid, gelaric acid, lactularic acid. The molecular weight is 1006 with low glass transition temperature of 36.4 degrees Celsius, making it quite interesting candidate for hot melt extrusion. It is having solubility. It is freely soluble in ethanol and slightly soluble in acetone, and it is completely insoluble in water because it is pH dependent soluble. Then we can see from the solubility in aqueous media, the problem of shellac is, the real challenge is it dissolves at pH 7, and it is completely soluble at pH 7.3 and above. So how can we try to use shellac and make it in a form that it can achieve supersaturation, maintain that supersaturation and achieve the solubility enhancement? This is what I will make you through. Then the next polymer I tried to investigate was afinisol, which was launched by DuPont in 2015. It is a HPMC based polymer, which is having very low glass transition temperature. And compared to the conventional HPMCs, which are having glass transition temperature above 150 degrees Celsius, making a processing challenge for hot melt extrusion. The DuPont came with an polymer having a glass transition temperature of 96.6 degrees Celsius. So we can extrude it below 150 degrees Celsius, and which is very good for the API, which gets degraded at higher temperature. Then I prepared some of the mixtures. In these mixtures, I investigated the binary mixtures, which was loratadine with afinisol. 20 stands for the API and 80 stands for the polymer, while another binary mixture was loratadine and shellac SSB55, which was same ratio 20-80. Then I tried to make some ternary mixtures to see that if we can achieve our goal. So loratadine, afinisol, and shellac were mixed together because we assumed that due to the shellac's low molecular weight, it has a higher tendency to get mobilized with HPMC, and it can form a uniform matrix, and that can be easily extrudable, as well as we can do the spray drying on it. So we prepared ternary mixtures in ratio like 20-75-5. So 20 stands for API, 70 for afinisol, and 5 for shellac. And we varied the ratio of afinisol and shellac in order to see that we can modulate the dissolution profile or not. And we prepared this by via turbula mixture for amorphous solid dispersion preparation, and also for spray drying. And for DSC and ATIR, we used ball mill, where we keep it using a rest ball mill at 30 hertz for 3 times in 5-minute cycle. And after each 5-minute cycle, the break was taken to minimize the thermal energy intake, which is quite common after ball milling. Then how to manufacture. The first case study was using shellac for spray drying. And in this, we try to do preliminary screening, which is pretty important for spray drying purpose. In a spray drying, you dissolve API and a polymer in an organic solvent or in a water combination, whichever it may be. The API and polymer should form an homogeneous solution. This is the most important criteria. And then you can subject it to the spray drying process, where the liquid is being sprayed into the drying chamber, and automation takes place, and there is a creation of fine mist, and this fine mist leads to the formation of a powder, and it leads to formation of from crystalline to amorphous form. And we did some solvent screening, where we found out that acetone and ethanol in the ratio of 70-30 helps to achieve homogeneous solution. So we use this solvent, and the solid content which I kept was 4.9 to 8%, and the octane yield was 43 to 45%. The set inlet temperature was 84 to 86, and the outlet temperature was 52 to 63, and aspirator was set at 90%, and the spray rate was 3 to 4 milliliter. So by that way, we obtained a binary and a ternary ASD. Then the second case was with the hot melt extrusion using shellac, same. So hot melt extrusion is widely known now in the pharmaceutical industry. It is a very evolving technique and very popular at the moment. So in what we do in hot melt extrusion is we make a mixture which I prepared previously. Then we have a feeder here, and from this feeder, the API, the polymer API mixture comes in contact with the conveying element here. So the drug and the polymer mixture are intensely mixed at a high shear rate, because we have in hot melt extrusion we have a barrel, which is the black line is for the barrel, and we are heating it barrel. So the melting and the kneading takes place in this inside the block, and it is conveyed via screws. We have a conveying blocks here, and this green stands for the kneading blocks, where the kneading angles are set at like 30, 60, or 90 or 0 degrees. So we can modulate that in order to obtain the right amount of shear rate. And it's also a trial and error experiment, and also it's based on experience of extrusion, where you can achieve higher shear rate, but you also have to take into account that you don't degrade your product. So you start with adding your mixture. The conveying block conveys your material, your powder feed. It comes into contact with the screw. Due to the heating, the melting takes place, and due to the friction between the screws, another heating is being exerted. So the melting and kneading takes place inside the block, and you can get from where you, after this there is a hole where that stands for the die. The filament comes out. You collect those filament, and then you can downstream it into a pellets or a powder or a granule or an implant. So by this technique, you can also get an amorphous solid dispersion. The basic rule of hot melt extrusion is you have to you have to analyze your sample and study first it for the miscibility studies, and the second thing is the glass transition temperature. So the rule of thumb is you extrude it always at 50 degrees Celsius above the glass transition temperature of your mixture. So based on my glass transition temperature, which I did for initially my physical mixtures, I try to extrude it above 50 degrees Celsius. So we used hot melt extrusion from 3Tech, which is a 12 millimeter extruder. We have 5 barrels into it. So for which stands for 5 heating zone 2. The first heating zone was for loratadine afinisol was 65, and the rest temperature was kept at 110 degrees Celsius with screw speed at 100 rpm. The torque was 7, and the feed rate was 1 gram per minute. The torque was higher in this case because afinisol is having a higher viscosity. So because of this viscosity, we can observe the torque, and because there will be exertion of we need a higher shear rate too. While in shellac, we can observe that the torque was low, which was quite observed from its property, because shellac is having low molecular weight and it is not viscous. So and also we try to extrude it at below 90 degrees Celsius, because shellac's glass transition temperature is 36.9 degrees Celsius. While the other mixtures of loratadine, afinisol, and shellac in the ratio of 20-75-5, we try to keep all the parameters constant and just record their torques. And we can see with increase in quantity of shellac, the torque got decreased. So we can say that afinisol, shellac got immobilized quite nicely with afinisol, leading to the decrease in the torque and increase in the molecular interaction. Then the evaluation of ASDs. We try to evaluate the ASDs on base of solid state via DSC. So using this is the graph of TOPM, which is a modulated differential scanning colorimetry, where you can heat your sample from -20 degrees Celsius for 2 minutes and keep it constant as an isothermal peak, and then again heat it from -20 to 160 degrees Celsius. And here we calculated the first using the first heating cycle, we got the glass transition temperature. With loratadine afinisol, the picture on the left side stands for the spread right ASDs, while on the right side it stands for the hot melt extrusion. With loratadine afinisol, we are the black one, we can observe the glass transition temperature 59 degrees Celsius with same observation for the hot melt extruded, while for shellac, the glass transition temperature was decreased and it was 30 to 40 degrees Celsius. While in case of other ternary mixtures, the glass transition temperature was decreasing based on the shellac concentration. So in case of loratadine, afinisol, and shellac in 20-75-5, the glass transition temperature observed was at 59 degrees Celsius, while in case of 20-40-40, the glass transition temperature decreased to 50 degrees Celsius. So it is indicating that the glass transition temperature, with the decrease in the glass transition temperature, the API is homogeneously mixed into the polymer matrix, leading to the increased interaction. Thus, it's a sign that they are miscible with each other. While after you get this the ASDs, we subjected it to also for checking an absence of endotherm, because if you have some small amount of API which is not dissolved or recrystallized, you can observe it in DSC. In our case, we observed that the API melting point was 137 degrees Celsius. In that point, no melting endotherm was observed, thus confirming that the amorphous solid dispersion are formed using hot melt extrusion as well as spray drying. Then another proof of conformation was XRPD. With XRPD, the black one stands for the loratadine neat, while the red and blue stands for afinisol and shellac, which are amorphous completely, while the physical mixture, pink and the rest is from afinisol and shellac, which we can observe that they are completely amorphous. In case of the ternary physical mixtures, we can clearly observe that the crystalline peak was appearing, which is comparable with the loratadine API, indicating that they exist in crystalline form. While in case of amorphous solid dispersion for the ternary mixtures, we can clearly observe that all the ternary mixtures were completely converted into amorphous form with with a halo pattern and no intense peak. While the same observation was with our hot melt extrudeds, in which all the samples were completely amorphous in nature. Then we try to do the ATIR in order to investigate the molecular interaction. The loratadine, it's a weak base. It is having it is showing a CO stretch at 1706, while afinisol, it is having higher OH groups, and it is observed at 3461.1 centimeter square. In shellac, we observed that the CO group was there and also the OH group. So the CO group was observed at 1706. While in case of the amorphous solid dispersion, the loratadine afinisol in spray drying and hot melt extruded, we observed the CO group was decreased at 1699, and the OH group was 3463. While in case of shellac, it was not that pronounced, so indicating that there is less amount of interaction. In case of the spray dried formulation for the ternary mixtures, we observed that in case of 2075-5, the OH stretch was decreased at 3447, while in case of 2070-10, it was decreased at 3446 compared with the API and the polymer mixture, indicating that there is a higher level of interaction. And since I made together all graph, it got squeezed. So the separately on the left side, you can observe one of my optimized ratio where the OH stretch was 3446 and CO stretch was 1696, indicating the increased interaction compared with the other ratios. Then we try to investigate the in vitro profile via dissolution using SOTAX. We used non-sync dissolution in order to observe the supersaturation behavior, which is not possible with the sync conditions. So the black line on the left side indicates the spray dried formulations, while the right side indicates the hot melt extrusion. The black one is the loratadine pure, where loratadine was 2 to 3% only dissolved at pH 6.8 for 3 hours, while the blue one is the loratadine shellac, having much lower dissolved amount because shellac is not soluble at pH 6.8, while the violet color here, which represents the loratadine shellac, which is 20-40-40. So shellac and afinisol is in the same ratio, but in this case only 10% amount of API was dissolved, while in case of loratadine in the ratio 20-75-5, we observed 20% amount of release, and which was comparable with afinisol in the as a binary mixture 2080, while in case of loratadine, when we try afinisol and shellac, we increase the afinisol concentration to 60%. We observe 40% amount dissolved at the at pH 6.8, while in case of loratadine, when we decrease the ratio of shellac and increase the concentration of afinisol, the higher amount of API was released, and which was attributed to the molecular increased intermolecular interaction. The green one is our 20, 70, and 10 percentage, where we observed 80% amount of the dissolved API, which were compared to 2%. So indicating that the solubility is enhanced and the supersaturation is also maintained. Then we try to calculate the solubility factor. The solubility factor is the improvement of the solubility for amorphous solid dispersion, which is related to the crystalline drug, and which is sees as in pure showed a solubility factor of 0.1, while the loratadine afinisol in spray dried and hot melt extruded showed like 0.7% solubility factor. So solubility was enhanced in 7 folds, while in shellac solid dispersions, which is binary one, it was only 0.2, which is 2 folds. In case of 5, the solubility factor was 2, so indicating 20 folds increase, while in case of 2070 and 10, the solubility factor was 3, so indicating there is 30 folds increase in the solubility, which was higher compared with the other ratios. Thus, we can observe here the solubility enhancement has been occurred to 30 folds, which is compared to quite higher with the loratadine pure. To conclude, loratadine ASDs with shellac and HPMC, the optimal ratio we found out was 20-70-10, showed a good control of supersaturation and improved dissolution behavior. This synergistic effect was due to the intense intermolecular interaction, which was qualitative confirmed by ATIR and was supported by our DSC results. And we think that this is due to the increased miscibility, improved wettability, which is leading to the observed dissolution studies. Thus, with the successful implementation of shellac with the ternary AST, we can achieve solubility improvement of loratadine. Thus, our current study will add to the understanding of using a shellac as a tweaker and additive in supersaturation occurrence and maintenance for solubility enhancement purpose, and it will add for future ternary ASD development for useful for solubility enhancement purposes. These are my references. I like to acknowledge my supervisor in Bonn, Professor Wagner, and then our principal, Chitlangi sir, Thomas ma'am, for inviting me, Nanaji sir, and Maruti sir for coordinating everything, and all the DY Patil IPSR staff. Thank you very much. Hello? Hello?
Hello.
Yeah.
Anjaman?
I guess some network issue at Anaga ma'am's place. We'll start with question and answer, sir.
Yes, yes.
So on behalf of students, I like to ask first question. So what is the scope and requirement for PhD abroad? I can see many of my master students. So I guess they like to know what is the scope and requirement for PhD if at all they plan after masters.
Well, first here is you should have a publication. The second criteria is to know also you have to make write a proposal for your PhD on topic what you have to work on, and you have to send it to the German professor. And your master's degree is valid in Germany, of course. You can apply it, but it's also difficult to get a funding. Yeah. So I think the criteria is very simple: is your qualification of masters, your good research work. The third thing is your passion, like what you want to really do with your PhD and your topic you like to work. So the proposal should be a good one, and the German professor should like it. And if everything is that goes well, then I guess it can work for you.
So in continuation with the same, so the master's work and in publication will play an important role, right, sir?
Yes.
Yeah. Yes, ma'am. Asha, ma'am.
Yes, yes. Dnyaneshwar, that was a very excellent session, and I would like to congratulate you for the excellent work you did in your PhD, and good luck for all your future endeavors in Germany. And at this point, I would also like to state that as an alumni, I think this is the first step that of your contribution towards the Alumni Association, and I think it is a need of the hour that students and researchers like you who are abroad should take time and interact with their juniors who are at the institute, because I'm sure that is going to be a great motivation for all these students, especially at DYP, who are listening to you today. Today actually you focused on two major techniques, spray drying, and then you talked about the hot melt extrusion. So as a researcher, can you just throw light on the current status of these two technologies, especially with their adaptation to the industry?
Yeah, so currently it's very popular in industry for new chemical entities, and also in India it's getting popular because of super generic concept. So to make super generic concept a little easier to understand, it's like you have different the product is already launched in market. It is an ANDA or it's an NDA, but you want to improve its solubility and get it into in better form. So some companies, they are trying to use hot melt extrusion or spray drying to increase their life cycle management. So industrially applicable, it comes under the paragraph of 505B filing of the United States FDA. In the current situation, the hot melt extrusion and spray drying, they are widely used because of the new chemical entities are evolving, and that's why I showed you the marketed preparation. Every year, a new amorphous solid dispersion is been approved by the United States FDA. The pharmaceutical companies are investing a lot because we have the problem of poor soluble APIs. The compound laboratory, the combinatorial chemistry leads to the poor solubility of APIs. You know this problem from pharmaceutical chemistry and drug discovery pipeline. So this address needs to be this problem needs to be addressed, and that's why spray drying and hot melt extrusion is very popular because of the scalability and because of its widely acceptance, and also it's taking much attention. If you check online for research articles, you can find that a lot of papers are published on hot melt extrusion and spray drying. So it is very industrial relevant. And in my sorry, current job also, I'm responsible for hot melt extrusion technique and spray drying. And you also know, ma'am, that in our college we had a spray dryer which I used in my masters, and that's why I got my first patent too. So it was a nice time there, and where our college indeed have some nice facilities. It's only, I know, compared to here, I will say the funding is the problem, and which will change in future, I guess. And of course, I believe that if we change our attitude, then we can change everything.
Yes, yes, very well said, right. Dnyaneshwar, one more question. As a researcher, can you just throw light on how, as a researcher, we need to make a judicious choice whether we go with spray drying or hot melt extrusion? So, what are the API profile characteristics that we need to take into consideration while we make the selection of these two techniques?
So each technique has its own advantage and own limitation. So for spray drying, first what I try to make is make a decision tree. So you have to check your API is soluble in organic solvent. Okay. Is your polymer soluble in organic solvent? Okay. Yes. In the same solvent, then it is fine. Is it getting super precipitated? That you need to check. Then the second thing involved is if you with hot melt extrusion, if your API is having higher melting point, so let's say above 150 or 200 degrees Celsius, then it is difficult to do the hot melt extrusion there. So we go for spray drying because it is how you say thermostable. It is better for thermolabile drugs, while hot melt extrusion is challenging. So hot melt extrusion, you need to take into account that your API is API and your polymer, they are miscible and they are soluble. So the miscibility means the API which is dissolved in amorphous form and the polymer which is dissolved in amorphous form. So this means miscibility, which is giving you 1 glass transition temperature, and the solubility resembles like your API is a solute and your polymer is a solvent. So the amount you increase, it should get dissolved. So it is the same like we do in our UV's calibration, where we increase the concentration of the API and it gets dissolved in the solvent. The same is with this. We need to assess this and we can do this via DSC. So based on these two things, we can get an idea. Oh well, it works there. So this API suits in hot melt extrusion process, and this API suits in the spray drying process. But sometimes it is also the problem that API is not soluble at all, and it is also very highly having high glass transition temperature and melting temperature. Then you have to go for another approach, which is called co-crystal approach. So the pharmaceutical companies are also working on this technique. So co-crystal is also one of the new coming up technique. And yeah, in my knowledge now, hot melt extrusion has a wide applications for making filaments, making 3D strands, 3D printing strands for personalized medicine. So the pharmaceutical companies are investing a lot at the moment on these technologies.
Dnyaneshwar.
Hello. Yes,
You can take other questions.
yes, ma'am. There is one question from Pranav Joshi. So he wants to know what is the importance of shellac towards understanding of its importance in vivo system.
Okay, it's a very good question. I think in what I understand from your question is he wants to understand if it's applicable in vivo studies. Is it right?
Yes, yes, sir.
Okay, so in my current study, I have not performed the in vivo aspects of shellac, but I'm not sure about it. But one thing what I know about shellac, it is a grass listed, so generally regarded as safe. So we can of course use it, and it is also dissolving at pH 7 and at 7.4. But yeah, it makes an very good approach in application. Only the limitation what I found with shellac, it is obtained from the insects lac. So I'm not sure about its toxicity profile, even though it is a grass listed.
Okay, thank you, sir. Sir, there is one more question from Pranav only. He wants to know whether this shellac increases the solubility of drugs which are used in the cancer therapy.
That's a very good question. My second paper is focused on that, where I used shellac for colon targeting, for colon cancer delivery, and shellac can be used.
Okay, thank you, sir. Any other participant who wants to ask anything can mute, they can unmute themselves and interact with sir.
Dnyaneshwar,
Yes, sir.
first I'd like to congratulate for your wonderful session. My question is, what is the scope of natural therapy in the treatment of disease conditions? Suppose from Germany point of view.
Well, at the moment, Germany is going more herbal. People are getting more vegan diet now, which is a good part. And Germany has a good reputation now for herbal things, and they are looking at quite constructively at the moment. The German government and Indian government have a good collaboration at the moment for herbal research, and they are investing a lot in this, and they have termed uh, one one thing which is called Ayurgenomics. So you use herbal or Ayurvedic medicines or herbs, and you apply genomic principles to them to fight with the disease condition. And this recent collaboration was done by Frankfurt Center for Biotechnology, which is based in Frankfurt, and with the Ministry of Ayush of Government of India. And they are working on this principle of Ayurgenomics, and it's a very nice collaboration. Actually, I can send you the links, and if you like, it's they are taking it very further. Actually, this natural things.
Hello. Yes, sir. I want to ask one question. What all criteria needs to be considered while selecting a university and guide for doing PhD in abroad?
Yeah, first is your research interest. Secondly is the uh, your publication, which is very important here, because then the third thing is the university's reputation, and what the professor is doing, it should match your profile. Then the fourth thing is you should first think, why I want to do PhD abroad. Does it make sense to go to abroad? Why can't I do it in India?
Okay.
So for me, why I went to Germany, the main reason was to learn about hot melt extrusion, because spray drying, I have already worked on it and I know about spray drying. So I worked in company also for 2 years, but I thought like, oh, hot melt extrusion is a very upcoming technique, and